there are two terms that relate to all this. One's called genotype and one's called phenotype. And in a nutshell, the genotype just means the alleles the organism has, and the phenotype means what physical traits the organism has. And the terms kind of make sense. The genotype is its genes, and phenotype are its physical characteristics. Or some people say the phenotype is what I can see with my naked eye when I look at the organism. And the genotype is what's inside it. It's the DNA and the alleles inside the organism. You can't see with your naked eye, obviously. Now, remembering that every organism has two of each gene, because we always have two parents, each organism is going to have two alleles. They might both be the same allele, or they might be the dominant or recessive allele in that organism. If the organism has two of the dominant alleles, in other words, if it's big P, you say it's a genotype. You can say big P, or you can say homozygous dominant. Homozygous means two of the same allele, and since big P is the dominant allele, you can say it's homozygous dominant. If the organism inherited a big P allele from one parent and a little p allele from another parent, in other words, it has one of the dominant alleles and one of the recessive alleles, you say that organism's genotype is heterozygous, or you can say it's big P, little p, that's fine also. If the organism inherited the recessive allele from both parents, you would say the genotype is little p, or homozygous recessive. Now, sometimes people call the homozygous dominant genotype true breeding. I don't use that term that much, but you sometimes see it in textbooks. And sometimes they call the heterozygous genotype, the big P, little p, as being a hybrid genotype. And again, I don't use that one too much, but you do see it in some textbooks. All right, and remember you can't see the genotype with your naked eye, but what you can see are the phenotypes, the physical traits that come from the genotype. And so if you were looking at plants, if this was the gene for flower color we were talking about, this is what the plants would look like. The ones that show the trait from the dominant allele are said to have the dominant phenotype. You can see it there and there. But the ones that show the trait from the recessive allele are said to have the recessive phenotype. Now notice a few important points here. Point number one is genotype controls phenotype, which you know because your physical traits come from the proteins that you have inside you. Your proteins carry out the tasks that run your body. But the proteins that you have come from what genes, what alleles you have. So in final analysis, it's your genotype that gives you, that creates your phenotype. The second point here is that notice you have the same dominant phenotype no matter whether you have two of the dominant allele or just one of the dominant allele. At first that seems a little weird. You think, well, if I only have one, if this, you might say, if this allele is making purple pigment, if it's an enzyme for making purple pigments, and there's only one of it, why isn't this a lighter shade of purple? Well, the cells can adjust how active a protein is, and if there's only one copy of that protein, the cells, the organism, will make more of that protein or make that protein more active to compensate for the fact that there's one copy of it. So you end up with the same amount of purple pigment. A good analogy is, think of your car keys. Here's my car key. I can have one of it, or I can have two of these car keys on my keychain, but my car doesn't run any different whether I have one or two. You get the same result whether you have two car keys or one car key in terms of how well your car runs. That's the way to think of dominant alleles. And a third point here is that when you look at an organism, you can see its phenotype. If you look at the organism and you see the recessive phenotype, you know its genotype because the only genotype that gives you the recessive phenotype is little p, all is recessive. But if you see an organism showing a dominant phenotype, you don't know its genotype for sure. It could be big P, almost like it's dominant, or it could be heterozygous, big P, little p. You're just not sure yet. And that concept, that organism showing the dominant phenotype, you don't know their genotype, it could be big P, or big P, little p, is important. When we start doing genetics problems, you'll have to rely on that concept to get the right answers on the genetics problems. So don't forget. All right, any questions up to here? All right, you guys are kind of quiet today. So one thing I started in on saying about 5 minutes ago that I got sidetracked is this, that when you're asked to do genetics problems, you could, in theory, draw them out like this, but this is not a really efficient way to approach genetics problems. After Mendel developed his laws of genetics, a systematic way of diagramming genetics process was worked out called Punnett squares. And this is the way you should approach genetics problems in this class. And here's what they look like. We'll use this as the crossing. This is the one where Mendel took that true breeding purple flower, big P, and a true breeding white flower, little p, and crossed them so that the F1 generation here was all big P, little p, purple flowers. All right, so if you're doing a Punnett square, first identify what parents you're talking about. This one's big P genotype, and this one's little p genotype. And then you draw a box like this, a square that's subdivided into 4 smaller squares. And what you do is write the gametes that this parent can make on that side of the square, and you write the gametes that this parent can make on that side of the square. Now in this case it's easy because each gamete will only get one of these alleles, because gametes are haploid, but it's both the same allele, so it's going to be big P, and big P is the 2 possible, the gametes that one can make. And the same with the white flower. Each gamete will only get one of those alleles, but they're both the same, so all gametes will carry the little p allele from that parent. And then what you do is you fill in all the squares in the table with the allele above it, and the allele to the side of it, there and there. So your Punnett square ends up looking like that. So what the Punnett square tells you is essentially what the genotypes of the offspring are going to be. And so all the little squares in the Punnett square table show big P, little p, and so this says all the offspring are going to be big P, little p. In other words, you're going to get purple offspring. And notice that that's consistent with what Mendel found. The F1 generation was all entirely purple, so it works, in other words. Let's do the F2 generation. So Mendel then took this F1 generation, which remember are all big P, little p, and he crossed them with themselves to see what the F2 generation would be. And he found generally of the F2 generation that was produced, 3 out of the 4 were purple, but 1 out of the 4 was white. Here's the Punnett square that crossing the F1 generation. So all the F1 generations are big P, little p. So to do the Punnett square, you draw the big square, subdivide into 4 smaller squares, and then next to this parent, you draw the 2 types of gametes it can make. Half of its sperm will carry the big P allele, and half of its sperm will carry the little p allele. And the same with this parent. When it makes eggs, half of them will carry the big P allele, and half of the eggs will carry the little p allele. Like that. And then you fill in the Punnett square, with whatever allele is above it, whatever allele is to the left of it. And so notice that this Punnett square gives us 1 little box that's big P, 2 boxes that are big P, little p, and 1 box that's little p. And when we say, okay, those are the genotypes, what phenotypes do we get? this is what you get. That the phenotypes produced, 3 out of the 4 were purple and 1 out of the 4 were white, and that's exactly what Mendel saw in his cross of the F1 generation. Here are the actual numbers. He got 705 purples and 224 whites, which are more or less a 3-to-1 ratio. All right, let's see, that's probably a good stopping point. We'll pick this up again on Monday, and put your stuff away, please, for your quiz on mitosis.